Welcome back to Bio 111. This is Chapter 15, Section 3, Eukaryotic Transcription. Alright, so in eukaryotes we have several types of polymerase. We have RNA polymerase 1. It transcribes rRNA genes. RNA polymerase 2 transcribes protein coding genes. And RNA polymerase 3 transcribes rRNA, tRNA, and small nuclear RNA genes. Alright, so look back on what these types of RNA are. rRNA, little r, rRNA, is ribosomal RNA. Ribosomes are made up of proteins, so enzymes, and RNA. That RNA inside the ribosome we call rRNA, just to give it a name so we know what to call it. Alright. tRNA, the T stands for transfer. These are pieces of RNA that specialize into bringing specific amino acids to the ribosome, and a specific tRNA only binds to a specific section of the RNA, a specific codon, and it only carries a specific amino acid. All right, and that means that if it's able to bind or be incorporated into the ribosome, it's going to bring in the correct amino acid to add to the chain that matches the mRNA. All right. All right. So in eukaryotes, we have something called a transcription factor. These are molecules that bind to promoter regions and begin the initiation of transcription, of mRNA transcription. All right. RNA polymerase 2 then binds and forms the transcription complex, the initiation complex. All right, so elongation, mRNA is synthesized from the 5' to the 3', like we saw earlier. Now remember, in eukaryotes, that's what we are. Plants, animals, fungi, the more complex organisms that have organelles like the mitochondria, they have nuclei, they have rough ER and Golgi apparatus. We have all these complex structures inside the cells. We also have lots of DNA. And if you remember from the previous classes, we talked about the way they pack, the way that eukaryotes pack their DNA. They use histone molecules to wrap the DNA like string on a spool. Right? We have to pull some of those spools apart, some of those histones apart. All right? These nucleosomes apart to get to the DNA that's wrapped around it so we can transcribe it. This is what we're seeing here. Now, once we transcribe that section of DNA, we've got to put that nucleosome back together. All right, that's what's going on here as well. All right, termination. RNA polymerase 1 requires a termination protein that recognizes a specific 18 nucleotide sequence. RNA polymerase 3 terminates transcription via hairpin formation. RNA polymerase 2 transcribes 1,000 to 2,000 nucleotide excess of the gene template, which are removed during mRNA processing. All right, one thing that's different inside of, and we're going to see a little bit later, between a prokaryote and a eukaryote. In a prokaryote, all of the information that you see in the DNA, that comes out in the DNA, is transcribed to the RNA, and we keep everything. All right? Eukaryotes are different. There's a lot of stuff inside of the DNA and subsequently inside the RNA that's not needed. We got to take out. Right? So there's some post-transcriptional processing that has to happen. All right? This is it for section 3. See the next video for section 4.